Hello, everyone, and welcome to this session of the GCI Summit. My name is Dr. Paul Gavin. I'm the Chief Executive Officer of Avecho Biotechnology. Avecho is a Melbourne-based company with a novel formulation technology called TPM. Now, our background is pharmaceutical drug development for traditional non-cannabinoid medicines. In this space, we've been using our TPM to increase the solubility and bioavailability of pharmaceuticals, and then conducting formal clinical trials to demonstrate their performance in people. Today, though, I'm going to talk to you about how TPM can be used to overcome a range of problems associated with the formulation of cannabinoids, specifically their poor solubility and bioavailability. So, look, to set the scene, in the last decade, there's been a huge shift in the treatment of medicinal cannabis following the recognition of the therapeutic potential of cannabis and cannabis extracts. Now, many countries around the world are currently enacting some sort of legislation to make medicinal cannabis legal for therapeutic purposes. Additional countries have gone further and are allowing markets in both consumer and recreational areas. But I guess from the biotech side, the fact that it's been illegal for so long means that there's really been limited R&D conducted into formulation development. And so initial product offerings to patients and consumers have traditionally been oil-based vehicles. Moving forward, a range of more acceptable dosage forms are going to be required to fulfill the needs of patients, physicians, and consumers. However, the cannabinoids themselves suffer from a range of technical issues that make formulation difficult. And so cannabinoids, which are the key active components of medicinal cannabis, are oil-soluble molecules with poor solubility in water. And like all other oil-soluble molecules, they have very poor oral bioavailability. So depending on the literature that you'll read, you'll see figures reported from potentially anywhere from 3 to 10% bioavailability for the cannabinoids. So what this means is that 90% of the cannabinoids in the product that patients or consumers are consuming is metabolized or excreted and is never absorbed to give any kind of biological value. So the low bioavailability presents real challenges for developing formulations that can deliver cannabinoids to the body efficiently. And so this has become a real focus of a lot of formulation labs around the world, because if you can increase oral bioavailability, you can potentially give greater therapeutic effect because there's more cannabinoid absorbed to exert a biological influence. You can potentially shoot for new clinical indications. So try and treat indications that ordinarily would require huge doses of the cannabinoids. You can reduce the cost to patients, which is a major impediment for them, especially in Australia. And on the commercial side, you can really provide some technical and commercial differentiation. And so this is really where Avecho has spent the last year investigating how our TPM may potentially add value to the cannabinoid space. And so what is TPM? This is my single slide overview of TPM. I'll go into a bit more detail shortly, but it's the core component of our drug delivery platform. TPM itself, it's a mixture of two different forms of phosphorylated vitamin E, which we manufacture at our facility in Melbourne. Now the TPM, because it's tocopherol phosphate, has enhanced vitamin E activity compared to normal tocopherol. And quite uniquely, when you put it into a system that contains water, whether that's a formulation or the stomach, the TPM can self-assemble into tiny vesicles that encapsulate drug molecules. And this encapsulation can increase drug solubility and stability, especially for drugs that are poorly soluble in water. So you'll hear me say that a lot in the next 20 minutes. Now, an example of that is this image that you can see here. So this is one of our non-cannabinoid formulations. This is a reformulated propofol injection. Propofol itself, the drug, it's an oil, it has no solubility in water. And so the commercial product, as you can see on the right, is an opaque lipid emulsion. Now, we reformulated it, adding a little bit of our TPM, a bit of our vitamin E, and created a transparent microemulsion, which you can see on the left. And so when we use TPM to encapsulate drugs, apart from solubility and stability aspects, it also allows us to increase our absorption, both in the oral route of administration, so oral bioavailability, as well as topical dermal and transdermal absorption. Now, it's built from vitamin E, so it's very safe. And to summarize, I guess, when we reformulate drugs with TPM, it allows us to give the products improved pharmaceutical properties and performance, and allows commercial opportunities for differentiation and patent protection. So in the next few slides, I've got a bit more detail for the formulation scientists amongst the audience. So this first slide here talks about the structure of the components in TPM. So as you can see at the top, we start with vitamin E, normal alpha tocopherol. Our phosphorylation reaction creates tocopherol phosphate, which is the structure you see on the bottom left. So it's just vitamin E with a phosphate group stuck on the end. And on the right, you see the other component of TPM, which is ditocopherol phosphate, which is essentially two vitamin E molecules stuck together by a phosphate bridge. And together, this mixture is called TPM. And this mixture always contains TP and T2P at a roughly 2-to-1 ratio, which turns out to be pretty important for a number of the applications that we use it for. Now, TPM has enhanced vitamin E activity. So we actually spent the first probably 5 years of the company life characterizing TPM as a form of vitamin E. The product was actually sold in the US on market as a vitamin supplement called Ester E. And consequently, we did a lot of work characterizing its activity. And so we published a lot and proved that TPM, or tocopherol phosphates, have enhanced vitamin E activity for a range of the non-antioxidant vitamin E roles. It has anti-inflammatory activity. It provides benefits for dyslipidemia. It can improve endothelial dysfunction. It's a potential protective agent for cardioprotection. And it's very good at dermal anti-erythema. And consequently, it's sold into personal care products all around the world, specifically around reducing dermal erythema. On the safety side, it's metabolized to tocopherol, and as I mentioned earlier, has self-affirmed GRAS status for oral consumption. And so I guess why do I mention this vitamin E activity? Because it really differentiates itself from other technologies, surfactants that you might typically use to increase absorption. And the interesting thing specifically about the TPM, it's a range of these properties that it has actually appear to be complementary for some of the indications that you ordinarily might use CBD or other cannabinoids for. So it might be that TPM can increase the absorption of the cannabinoids whilst also exerting its own additional benefits. Now, the unanticipated consequence of phosphorylating tocopherol was that it would now form vesicles, self-assemble into these tiny little nanoparticles that are less than the width of a human hair. In retrospect, it's probably not that surprising because we've taken, as you can see on the top right, a structure that has a big water-hating element on one end. We've put a water-liking element on the other end. And so consequently, we've made a molecule that structurally looks a little bit analogous to a phospholipid, which everyone knows makes liposomes. And so as you can see in the images on the left, this is what a TPM colloidal suspension looks like. As we increase resolution all the way up to cryo-TEM, you can see a highly ordered vesicle structure made purely out of TP and T2P. And the important thing about these vesicles is that they encapsulate and solubilize drug. And again, it works particularly well with lipid-soluble molecules. So again, the image you see on the left, it's an example of a non-cannabinoid. This is a vitamin K injection that we've been working on. Again, vitamin K, it's in soluble oil. And on the image on the left, you can see that without TPM, vitamin K just sits as a non-dispersed lipid phase on the surface of the vehicle. However, when you add TPM, it's no longer on the surface because it's been dissolved and encapsulated by the molecules and evenly distributed throughout the formulation. Now, this encapsulation experiment, just this with and without TPM, is usually what we start with because it gives us a pretty good idea if a drug's going to plug directly into our technology. And when we can prove that TPM encapsulates drug, history shows that there's a range of advantages this can bring for other dosage forms and routes of administration. And so we've shown that it will increase oral bioavailability. So with this route of administration, when you co-dissolve TPM in an oil carrier with an oil-soluble drug and they hit the gastric space, the mechanism of action that we're sort of examining at the moment is that TPM vesicularizes and emulsifies the lipid, very similarly to the way that the gastric system works anyway. And so it may be that TPM shortcuts this process or allows it to occur more efficiently for subsequent absorption. It also allows increased topical absorption. So the TPM itself absorbs quite well into the strata corneum. It can carry drugs deeper into the dermal layers, and we've done a lot of clinical trials on transdermal and dermal absorption of molecules using TPM. And on the injectable side, it allows you to replace pretty nasty surfactants, chromophore, these kind of things, with essentially vitamin E. So as you see on the image on the left, that's actually an example of injectable formulation. And so I've just got 2 slides here demonstrating data on this increased absorption. Now, this first slide is around low bioavailability of lipids. And in this case, we've got a model drug here, which is coenzyme Q10. Now, on the left, what you're looking at there is solubility and dispersibility of coenzyme Q10 in in vitro gastric absorption model. So aqueous system meant to mimic the contents of the stomach. And as you can see, CoQ alone, which is the black line, it has pretty poor solubility in the gastric space. It stays poor if you put normal vitamin E or vitamin E acetate into the system. But if you add TPM, you can see suddenly that the solubility of coenzyme Q10 in the gastric space is increased. Now, when you turn to the graph on the right, this is what happens when you give those same formulations to animals and monitor the blood concentration over time. And what you can see, again, comparing the blue line, which is the TPM CoQ formulation versus all the others, you get a significant increase in bioavailability. And this has been true for other lipid-soluble molecules that we've looked at as well. On the topical side, you get a similar kind of phenomenon. So this is, again, animal experimentation. We've kept it constant using CoQ as this example, although we've done a range of drugs and pharmaceutical products, both in animals and human clinical trials. And as you can see, when you add CoQ alone to the surface of the skin, animal models, there's very little absorption. But when you add it with TPM, a formulation with TPM vesicles, you can see a significant increase in the amount of coenzyme Q10 that's absorbed into the skin. Now, the graph on the right is the area under the curve of the plasma concentration. So the amount of drug delivered transdermally in the same experiment. And you can see, again, when formulated with TPM, there's a significant increase in the amount of transdermal coenzyme Q10 delivered. And so you can probably all see where this is going already. So we have a problem with cannabinoids. They're oil-soluble drugs. They have poor solubility. They have poor bioavailability. We have a vitamin E-based technology that allows you to increase all these things. And so, I mean, from our perspective, if TPM did for cannabinoids what it has done historically for all other lipids that we've examined, it'd provide us with an opportunity to develop, really differentiated cannabinoid formulations that perform better than the first generation products that kind of entered the market. And so where do we begin? We begin, as I've discussed, in the simple proof of concept study, will TPM dissolve cannabinoids? And so as you can see with the image on the left, it clearly does. So the vial on the right is CBD. CBD just introduced into this aqueous system. You can see it's insoluble. It's precipitating, falling out of solution everywhere. Whereas the image on the right with TPM, there's no solid precipitate, there's no separated oil because the CBD has been encapsulated in the nanoparticles and evenly dispersed throughout the formulation. And as we discussed previously, this allows us to look at a number of different dosage forms and routes of administration. And so where we began was looking at whether this encapsulation led to the increased oral bioavailability of cannabinoids and also whether it would work topically for transdermal products. I should just quickly say this encapsulation experiment, again, because it's a lipophilicity game, we've done this proof of concept study using synthetic CBD, natural CBD, THC, both synthetic and natural, and all full spectrum isolates. And so the TPM encapsulates the cannabinoid extracts equally well, irrespective of their source. And so the first data to look at is the increased oral bioavailability of CBD. So we really undertook to try and learn how we could optimize a ratio of TPM to cannabinoid, as well as the nature of the other excipients to maximize the oral bioavailability. And this work, we did a lot of it in Denmark. And simply put, we ran through in vitro digestion models, exactly as you saw with the coenzyme Q10, to try and optimize some of the formulation parameters. And then we took the formulations across into animal studies. And so the data that I'm presenting here today is really the data from the animal studies. So this is rat PK. The graph on the top is the area under the curve, so the total amount of CBD absorbed over time. The graph on the bottom is the Cmax, so the peak CBD concentration during the experiment. All of the animals had the same amount of CBD dosed, and everything was compared against a control formulation. Now, this control formulation is CBD at 100 mg per mil in MCT, very similar to how patients in Australia are receiving the CBD under prescription. Now, you can see, as reported in the literature, when you give CBD in MCT, there's really not a great amount of absorption from that formulation. Now, when we add a little bit of TPM, which is the one labeled TPM1, to that same MCT formulation, you can see that the oral bioavailability goes up by about 4 times. When we add a little bit more TPM, which is the next formulation along, number 2, the increase in bioavailability goes up about 12 times. And the last formulation number 3, the increased amount of CBD absorbed from this formulation was around 40 times. Now, very much going to caveat that and say that we don't expect for 1 second we'll get a 40 times increase in CBD in people compared to the control formulation. But that's the way the results have come out from this animal study, and it allowed us very confidently to look at formulation TPM3 and say, this is the formulation that we want to take forward into further development. Now, on the topical side, what we did is that we took one of the commercial formulations that we developed and it's on market, and that's this Voverin TPM formulation that you see here. So this is a formulation which is currently sold by Novartis in India, and we optimized it for the delivery of diclofenac. So this is an NSAID local pain formulation. And we took the diclofenac out of the product and we replaced it with CBD to do some initial experiments where we monitor drug delivery into the skin. And that's what we're looking at on this graph here. So the 1% CBD formulation, which is the 2 bars on the left, is really this Voverin TPM formulation backbone. You can see that in the presence of TPM, the amount of CBD absorbed in the skin goes up by about 3 times. Now, in a different formulation vehicle that we'd also optimized for another topical application, you could get the CBD up to about 4 times in the formulation, and you can see that the presence of TPM increases drug absorption by 5 times. So the TPM plays a really good, strong role in increasing the absorption of CBD into the skin. And given what we know as well about TPM's effect on the skin and dermal indications, it looks like a pretty interesting concept for some of the dermatology indications that we're interested in. So what are we doing now? I mean, we've got this technology. We've shown that we can optimize some formulations. We really wanted to assess whether these TPM formulations would perform well in human clinical trials. And so we put this program together. I've talked about some of the data already. We've done our proof of concept where we showed that TPM could encapsulate cannabinoids. Independently, that was actually verified in the US by Purises. We did formal formulation development to optimize the ratio of the TPM to the cannabinoid to the other ingredients. We did preclinical studies to confirm increased bioavailability. And now we're now going into human clinical trials to test the effect of this product. And this product now for us has an overarching aim of being registered with the Australian TGA. So the fill formulation that we were testing in Denmark, we've now given that to Catalent. Catalent are doing all the GMP work to turn this into a soft gel capsule, 75 mg dose per capsule. This you'll understand a bit. I'll talk about the TGA in a moment. I'll talk about the TGA now. So the TGA is Australia's version of the FDA. What they did late last year is they issued a new guidance that CBD, pure CBD, could be prescribed over the counter. So you wouldn't need to go to a GP, you wouldn't need a prescription. You could just go into the pharmacy and get CBD directly from the pharmacist. But it had 2 caveats. The first was that it had a maximal daily dose of 150 milligrams. And the second caveat was that it was registered. It was a registered product, which means demonstrated safety, demonstrated efficacy for a given indication. And so a 75 milligram dose would allow 2 capsules per day to fall within the 150 milligram threshold that our regulatory agency has mandated. So the fill formulation itself, the oil that we developed, is now on an observational clinical trial in Australia. The soft gel capsule itself, when it's finished its GMP campaign at Catalent, we'll go into formal phase 1 PK studies in Australia, which will then be followed by our pivotal phase 3 studies, which will support the registration. So, I mean, the cannabinoids are a new space for us. And the interesting thing about it compared to the other pharmaceutical products that we've worked on is, whilst we're obviously spending all the money to take something towards pharmaceutical registration, it's the first time we've worked on something that could, in parallel, be sold into other geographies, consumer markets, other sectors to really allow the product to be commercialized much earlier than it would ordinarily. And so we're currently exploring partnerships around the formulation for other geographies. And so in summary, what we've shown today, I guess, I hope that we've convinced you that we've got a formulation technology which is built from vitamin E that increases the solubility of cannabinoids, specifically CBD in some of these examples. It's applicable to a broad range of dosage forms. So, I mean, injections, intranasal, patches, gels, creams, oral oils, tablets, capsules. We've kind of looked at them all over the many years. It increases the oral bioavailability of CBD, which is a real limitation for CBD and the other cannabinoids currently on market. And it also increases topical absorption as well. The interesting thing for us is that the TPM itself has complementary activity for some indications. We're currently exploring what indication would specifically benefit from a combination of TPM and the API, even though from all our regulatory work, we still are treating TPM as an excipient. And just the products that we have under develop are these soft gel capsules. We've got an oral oil as well, topical formulations. And we're really moving these towards commercialization in the Australian market, but equally happy to consider other geographies as well. And so, look, I'd just like to thank everyone for taking the time to view our presentation today. Please make sure you tune into the other sessions of the GCI Summit. If anyone has any questions about the content of the presentation today or about the technology or the products that we've been working on, please message me directly on the networking app. And yes, again, thank you very much for your time.